welcome to this explainer. Today, we're embarking on a real investigative journey. We're diving into the critical forensic science that literally determines whether a new chemical compound becomes a life-saving medicine or, well, a multi-million-dollar failure. So, let's jump right into this fascinating world of pharmaceutical development.
Look at these staggering odds on the screen. Out of up to 10,000 screened compounds, only a single new drug actually makes it all the way to regulatory approval. Just one. This immense needle-in-a-haystack bottleneck makes our initial screening process absolutely vital. I mean, we simply cannot afford to take the wrong chemical candidate into full-scale clinical trials. The stakes are just too high.
And that is exactly why preformulation is our crucial first filter. Think of it as the systematic interrogation of a compound's inherent characteristics, long before any actual formulation even begins. We're looking at the physical, chemical, and analytical properties of the molecule to really understand its intrinsic blueprint. It's the essential bridge between a raw chemical discovery in the lab and a viable, safe medical product. By breaking down these properties systematically, scientists build a comprehensive profile that dictates literally every subsequent manufacturing step.
To get there, here's our scientific roadmap for today. We'll hit core physiochemical concepts, explore the power of polymorphism, unpack analytical instruments and methods, look at solubility and pH profiles, and wrap up with essential revision takeaways.
All right, phase 1. Isolating and understanding that intrinsic physicochemical blueprint of our drug substance.
The whole point here is predicting the future. By evaluating properties like stability, solubility, and excipient compatibility right out of the gate, we can actually predict and prevent catastrophic formulation challenges down the line. This isn't just some academic exercise, It supports the design of an optimal dosage form and saves massive amounts of development time and capital. We're stopping doomed formulations before they even start. Understanding stability ensures the final product remains effective and safe on the shelf, protecting the patient and the massive financial investment required to get it there.
Moving on to Section 2, we're zooming into the atomic level to examine polymorphism, where one exact chemical can have vastly different physical identities.
These visuals brilliantly capture what I'm talking about. On one side, crystalline solids feature highly ordered, tightly packed repeating atoms, like a perfectly built brick wall. On the other hand, amorphous forms have these totally randomly arranged molecules. Because they lack that tight, orderly crystal lattice, amorphous forms often yield a much higher solubility and a super rapid dissolution rate. Basically, that molecular chaos makes it so much easier for solvents to penetrate and dissolve the compound.
Now, we have to make a critical distinction here between the internal atomic architecture, the polymorph, and the crystal habit. The habit is merely the external, macroscopic geometric appearance. It's just the shape, like whether it looks like a needle or a prism. The internal polymorph is what dictates the core chemical behavior, like solubility and stability. The habit mostly just affects how the powder handles on the manufacturing floor, like its flow and bulk density.
And the real-world consequences of getting this wrong are immense. For instance, take the antibiotic novobiocin. It is completely inactive if you administer it in its crystalline form, no effect whatsoever. But in its amorphous form, it absorbs rapidly and gives a great therapeutic response. Crazy, right? Conversely, paracetamol actually has terrible compression properties in its most stable form. It literally has to be engineered into a specific, metastable polymorphic state, Form 2, just so it can survive being compressed into a solid tablet without shattering under the intense pressure of the press.
These structural differences also heavily dictate thermodynamic stability. You have drugs like metalazone that exhibit monotropic polymorphism, meaning they stubbornly hold on to just one stable form across all acceptable temperatures. But then you have a drug like carbamazepine, which is enantiotropic. It's basically a shapeshifter, changing its stable polymorphic identity depending on the exact thermodynamic environment or temperature range it's exposed to. We have to understand these shifts to prevent a drug from suddenly changing its physical state while just sitting there on a pharmacy shelf. All right, Section 3. Let's crack open the forensic toolkit and look at the analytical instruments we use to track all this down. To uncover these hidden molecular behaviors, we rely on a highly precise thermal toolkit, broken down here. First up, thermogravimetric analysis, or TGA. This is essentially watching the scale. It tracks weight changes to detect moisture loss or chemical breakdown as things heat up. Then there's differential thermal analysis, DTA, monitoring temperature differences between our sample and a reference. But the absolute crown jewel is differential scanning calorimetry, or DSC, which measures the exact difference in heat flow. DSC lets us pinpoint critical thermal events, like glass transitions, crystallization, and melting points, with unbelievable accuracy, giving us a complete map of the drug's physical boundaries.
So we know the melting point, great, but that's not enough, is it? We still have to answer a very practical physical engineering question. How do these microscopic, sometimes highly unpredictable particles actually flow through high-speed manufacturing machinery to reliably become a solid, uniform tablet without jamming up the whole press? Well, to measure that compressibility and flowability, we turn to Carr's index. It's a really simple but incredibly powerful piece of math that compares a powder's tap density against its bulk density. This ratio gives our formulation scientists an immediate, highly accurate prediction of how that powder is going to behave when it's subjected to intense industrial processing forces.
Using this metric, we can actually predict our manufacturing success before we even turn the machines on. A Carr's index between 5 and 15%, that guarantees excellent flow on the line. You're golden. But if your calculation yields anything above 40%, that is a massive glowing red flag. It warns us of an extremely poor, highly cohesive powder that's just going to stick to the equipment, jam the machinery, and completely ruin the dosage uniformity of the final medicine. Similarly, we calculate Hausner's ratio by dividing the tapped density by the bulk density. Think of this as our vital secondary double check for the powder's flow dynamics. It confirms the predictions we made with Carr's index and ensures the powder is genuinely 100% ready for mass production scale-up. And the thresholds here are incredibly strict. A ratio under 1.25 means you've got good flow. But if your Hausner's ratio climbs above 1.5, the flow is inherently so bad that even dumping chemical glidants into your powder mixture isn't going to save it. At that point, there's no Band-Aid fix. The entire particle engineering strategy has to be completely reevaluated.
Section 4. Okay, we've survived the manufacturing plant. Now for the real test, surviving the human body. Let's look at solubility and pH profiles.
Once a drug is actually inside the human body, it faces a gauntlet. The constantly varying pH levels of the gastrointestinal tract directly dictates the ionization state of the drug. Here's the catch. If a drug is ionized, it dissolves beautifully in water, but it really struggles to cross those lipid-rich cell membranes. If it's unionized, it crosses membranes super easily, but it might just refuse to dissolve in the stomach fluids in the first place. This delicate, high-stakes balance between solubility and permeability completely controls the ultimate therapeutic efficacy of the drug.
To master that balance, we rely on the Henderson-Hasselbalch equation. This formula lets us calculate the precise pKa of the drug. Think of the pKa as the exact tipping point, the specific pH where exactly half of the drug molecules are ionized and half are non-ionized. Knowing this lets us predict with pinpoint accuracy exactly where in the digestive tract our drug will be best absorbed. Or it tells us if we need to formulate it as a specific salt to artificially boost its solubility profile.
Finally, because human biological membranes are lipid-based, think oil and water, we have to measure the partition coefficient, commonly known as log P. By testing how the drug distributes itself between an oil phase, like octanol, and an aqueous, water phase, we determine its lipophilicity. A score greater than 1 is what we want. It proves the drug is lipophilic enough to successfully cross lipid cell membranes and actually enter the bloodstream. This is literally the final, ultimate hurdle to ensure the drug reaches its systemic target and delivers the cure.
And that brings us to Section 5. Let's lock in the most critical findings from our investigation with some high-yield revision takeaways.
So keep these high-yield facts front of mind. First, preformulation is the essential filter that stops costly downstream failures in their tracks. Second, polymorphic atomic structures strictly dictate a drug's bioavailability. Remember paracetamol and novobiocine? Third, thermal tools like DSC are what expose hidden material instabilities. Fourth, metrics like Carr's index perfectly predict if we can actually manufacture the stuff. And lastly, pKa and lipophilicity are the gatekeepers that determine cellular absorption in the human body. As we wrap up this explainer, I want to leave you with this thought. Out of the next 10,000 chemical compounds discovered in labs around the world, what hidden microscopic property, maybe a random amorphous shape, a slight shift in pKa, or a really stubborn crystal habit, will be the sole difference between a completely forgotten powder and the world's very next medical miracle? Thanks for exploring the incredible blueprint of drug discovery with me.